Open for you to see. Let me begin recording and start sharing the screen. I never shut those down, so you can always go back and see those. The only thing I shut down is the answers to the quizzes, and I'm not even sure if I've displayed those. Let me go look at that briefly.
They're typically available like a day or two later.
Yeah, no, normally the quizzes that are multiple choice, see, I don't have quiz 2A answers up here, so let me display that. Uh, but I'm talking about the essay or short answer ones. I have to go in there and literally, what does that mean? Oh, that one I got a date there, so that one's no longer available. I gotta get this out of the way. I don't look like I have answers for quiz 3A. I'll have to make a note of that and take a look and see if I've got answers of that. And there's the quiz 4A. I'll make that available to you. So for the other quizzes, usually 2 days after you take the quiz, I allow you to see the correct answers, and then that's displayed for a week or two. It says it on the quiz. While I'm here, let's turn on the genetics. And does anybody know, did we have the last lesson, the notes up? Chapter 5, was that the last lesson before? No, there's chapter 6. There it is, let's turn that on. If I don't have something available to you, you may need to send me a reminder. Like chapter 6, I forgot to turn it on. What I usually do is I ask students in the lab to let me know. And in past terms, they let me know, and then I turn it on. And I'm afraid I've gotten a little bit bad about checking, so, all right, there, I've got the answers to quiz 4A up. You haven't taken quiz 5 yet, have you? I think that's on chapter 8, so you haven't taken that one yet. All right, so
you could show us the answers to that if you want.
Yeah. Let me make a note. I need to check for quiz 3A. If you know, that the answer is up, say it. I'd have to go look into it. I don't want to do that now. All right, let's go into the lab. I'm sharing the screen, right? So we're going to be talking about Lab 9 today. Remember, you should be working on your unknown work. You have to finish that by around March 9th, and then turn it in, and it's going to be due on March 13th. And some of you haven't begun at all on your unknown work. And to tell you the truth, you're going to have to work like crazy to get it done. It does take time doing your unknown project. And you're going to have to work really quick because you haven't gotten started. Those of you who have gotten started, you're probably fine. But don't let it slack. I know that a lot of people were working on your infectious disease paper, and so you weren't working on your unknown work. And You do need to be working on your unknown work. And remember what you're doing in your unknown work. Let's see if I can show it to you. I was just there. Is put it in your unknown notes, which you can find under week 5 of the lab. It's also under the later week, like week 10 or something, because that's when it's due. So under week 5 lab, there's the information on the unknown report, and you're going to be recording everything in your unknown notes pages, including what your interpretations are, what you think you might, the possibilities are. You're going to put on the end of that page what test you'd like to run next and why you'd like to run it. And then when I send you the results, which I can't show that here, but I will put it in the comment section over here on the right. Let me see. Is there anybody here that doesn't mind my showing them the class, their unknown notes page? Like Courtney, Nicholas, Lucinda.
No.
Jenny. Okay, who is that who's speaking? Denise?
Courtney.
Courtney. All right, let me, I think I should stop sharing until I get Courtney here. Because I'm not sure what'll happen when I go through. I probably have to go through one at a time. Let's go in.
It was pointed out to me that I've put the wrong dates as far as the year goes on the notes.
The wrong date and the wrong year. That's fine. All right, I've got it here. Let me. Where the heck? There it is. Sorry, just getting a little confused here. Share the screen. All right, so you should be able to see Courtney's unknown notes now. Courtney Swidler, and she made a submission on February 16th. And I, on the right here, have comments. And the last one I did was February 19th. I sent her the capsule stain, which was the last test she asked for. Starting the capsule stain. So I'm actually up to date on this one. And the point is, I will, on the right, give notes as well as the results. Like this is the PR mannitol test. There's the capsule stain test. And she is doing correct. She's on the left submitting things, and you take a look. She submitted more than once. She submitted 2 times. And she's got February 10th. If the date's wrong, it's fine because I've got a date up here. But
It's just the year that's wrong. It's just the year that's wrong.
Oh, not a problem. Yeah, I see that's wrong. The gram stain, she got the results. She said that hers looked gram positive, and the shape was cockeye with possibility of tetrad, staphylococci, diplococci, 11 possible species that she's considering. The gelatin hydrolysis test reduced that to 4 possible species because it's gelatin positive for gelatinase. And then the fermentation, positive for acid, no gas. And she's saying it's reduced it to 1 possible species. That's really good if you got it reduced to 1 that quickly. But then she did the mannitol test, and that was acid, no gas. I'm not sure if here she says it's either Staphylococcus or Streptococcus, and then she did the capsule staining to differentiate between Staphylococcus and Streptococcus. Okay, and she doesn't have the results yet put up for the capsule stain, but I sent her the results. Okay, any question about any of that? All right, so let's go to lab 9. Oops. Come on. I gotta bring that just a little way so I can reach that. So grab 9, IDing gram-negative bacilli. There's some reading to do beforehand. Upon successfully completing this lab, you should be able to interpret the results for the following biochemical tests. IMVIC, which stands for indole methyl red Voges Prescure, and that's so complex that most people just say the VP test, and the citrate test. Carbohydrate fermentation, which this is more than one test. That could be glucose fermentation test, lactose fermentation test, mannitol fermentation test, and inositol or inositol fermentation test, and one other sugar which I've forgotten. So we've got 6 different carbohydrates you could test in our lab. There's the motility test and the hydrogen sulfide production test. Explain how to interpret the TSI, the triple sugar iron slant, which is how we're doing the hydrogen sulfide test. The SIMS deep tube, which also gives hydrogen sulfide, but it gives sulfur for S, I, indole, M for motility. So that's what SIM stands for. And then growth on McConkey's and HE or hectone enteric agar plates. They're used to look for lactose fermentation of gram-negatives, meaning both of these are selective and differential media that grow gram-negatives, and then you can distinguish differences between those that ferment the sugar lactose from those that do not. And then use biochemical analysis to solve case studies involving gram-negative bacterial infections. So there's a number of case studies. We're giving you some of the tests and the gram stain. Is there anything else we give you? Maybe something else. And some information about the patient who came in with the infection. And then you're supposed to take that information and ID what species is causing the infection. If you give the disease name, like tuberculosis, instead of Mycobacterium tuberculi, or tuberculosis, that's fine, but like I said, you should be narrowing it down to one species. If you just say something like gram-negative enterics, that's the whole family, and that would not be narrowing it down enough. Any questions about that? So on the case study, you're supposed to narrow it down to one species that's causing the infection. All right, so we're going to be looking at the members of the family Enterobacteriaceae, and the short name is enterics. They're all small, gram-negative, non-spore-forming bacilli, meaning they don't form endospores. They are facultative anaerobes. There are 53 genera. 24 genera have been known to cause human infections. So realize that this is a large family with over 100 different species and 53 genera, the genus. There's several species in a genus. But there's 24 genera that cause human infections. Below are the 12 most common genera that cause disease. They're all glucose fermenting oxidase negative organisms. So all of the Enterobacteriaceae that cause human disease ferment glucose and are oxidase negative. Most of these genera, which are listed above, Salmonella, Shigella, I know you've heard of, E. coli, or Escherichia. Most of these genera are normal microbiota of intestinal tracts, aka coliforms. Some of them are of people, like Escherichia. Others, like Salmonella, are coliforms of birds. They are opportunistic pathogens in that they cause urinary tract and wound infections, pneumonia, and septicemia, and possibly more. Common genera are responsible for causing opportunistic infections, including E. coli, Enterobacter, Klebsiella, Citrobacter, Serratia. Several of them cause gastroenteritis, like E. coli, and you get them commonly by consuming contaminated food products. It includes organisms that are human pathogens, like the genera Salmonella, Shigella, and then E. coli serovar 0157H7, meaning this is a bad variant of E. coli, and frequently when people get it, they get sick from getting this E. coli. The only difference is it's a different strain from the normal E. coli strains. Pseudomonas aeruginosa is similar to the organisms in the family Enterobacteriaceae in that they are gram-negative bacilli. They don't produce endospores. However, Pseudomonas does not ferment glucose, and it is an oxidase-positive bacterium. So this is one way, or I guess two ways, to separate the members of the Pseudomonaceae family from the family Enterobacteriaceae. Pseudomonaceae do not ferment glucose, and they're oxidase positive. The Enterobacteriaceae ferment glucose, and they're oxidase negative. Pseudomonas aeruginosa is an important opportunistic pathogen causing infections, particularly in immunosuppressed individuals, burn, and trauma patients. Pseudomonas aeruginosa also is an important healthcare acquired infection, which they're abbreviating HAI, and we will talk about that as a nosocomial infection, meaning that's two terms meaning the same thing. Healthcare acquired infection and nosocomial infection mean the same thing. It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli cultivated from clinically derived specimens. In this lab, we're going to look at the common biochemical tests used to identify gram-negative bacilli. All right, the first test we're going to look at is the indole test. Some organisms produce the enzyme tryptophanase that converts the amino acid tryptophan into the products indole and pyruvic acid. After overnight incubation, Kovacs's reagent is added to the tube, and if indole is in the tube, the Kovacs reagent will produce a red color at the surface. E. coli is positive for indole production. Klebsiella is negative, and negative looks yellow. This is supposed to be red, but in this photograph it looks kind of purple, but we'll take that as positive for the production of indole. Okay, and remember, tryptophan in the presence of tryptophanase produces indole. The next test we're going to look at is the methyl red and the Voges Proskura test. These are two tests that are frequently ran together, and you inoculate one tube and then you split it into two tubes, and you can run the methyl red test in one of the two tubes and the VP or Voges Proskura test in the second tube. Now realize that when you're doing the unknown test, you have to ask for either the methyl red test or the VP test to follow dichotomous logic, because if you're using dichotomous logic, you're only requesting one test at a time. But frequently these two are set up at the same time using one tube, and then it's split into two, and then we test for methyl red in one and Voges Proskura in the other. Methyl red, or the MR test, is looking, actually both of them, and the VP test, they're looking for the end products formed as a result of glycolysis. Acid products are made by the methyl red test, and then the neutral products are made by the VP test. So glucose is converted by glycolysis into pyruvate, but some of the end products can be acid products, and that is what the methyl red is looking for, the production of acid. The VP test is looking at the same, but instead of glycolysis making acid product, it's making neutral products, and the VP test is testing for that. And so both of them are looking at the metabolism of glucose. The VP test takes 2 days. The methyl red test, if you read about it, it's best to do it at both 2 days and 5 days. Generally 5 days is good enough. And so when you do your test, I will wait until 5 days after you request the methyl red test for the incubation period, and then send you the results after 5 days. And that's something else I meant to tell you when I was talking about the unknown notes pages. When you request a test, you have to go look up on the test how long that test has to be incubated, because you're not going to get the results until the time after incubation. And the methyl red test is one of the long ones. It takes 5 days. The longest is the gelatin test. It takes a week, and that's one reason why I gave you that one up front, because that one takes so long to run. All right, so the methyl red test is very simple. You simply incubate the tube, split it into one tube for methyl red, one test for VP, which I don't have shown, and then you add methyl red to the tube, and if the tube is acidic, the methyl red will turn the tube red. So this is methyl red positive, this is not red, and we've added methyl red to this, it's yellow, and so that's, this is methyl red where we added the methyl red to it, and it's negative because it's yellow. The VP test is a separate tube, and when we look at it, it also is red when it's positive. You add the reagents to that, you can read on it, there's actually 2 agents you have to add to it, and then incubate it after you add oxygen to the tube, and it turns red if it's VP positive, looking at neutral products made, and a negative, it'll be yellow. Any questions about MR and VP test? If not, let's move on to citrate utilization test. The citrate utilization test is looking if the bacteria has the ability to use citrate as its only carbon source. So this tube has citrate as the only carbon source in the tube. It does have a nitrogen source, and the tube, if the bacteria can utilize citrate and the nitrogen source, will turn blue, and so that'll be positive in the citrate test. If it can't utilize citrate and the nitrogen source, it won't grow and it will be green, and that's negative for the citrate test. And citrate is looking at the production of citrate in the Krebs cycle or the citric acid cycle, shown here. All right, the carbohydrate fermentation test or the carbohydrate metabolism test is looking to see if the bacteria can utilize one of the sugars in the tube, and each tube only has one sugar in it, and I think I gave you a lactose fermentation test. So you had lactose in your tube, and then you look to see if your unknown bacteria could ferment that sugar lactose. If it ferments the sugar lactose, acid products will be built up in the fermentation, and then the tube will shift in color from pinkish or orangish color to yellow if it's positive in the fermentation test. Now, in the carbohydrate metabolism test, also called the carbohydrate fermentation test, you could get fermentation without the production of acid, so there's no bubble in the Durham tube, or you could get acid produced with a bubble. And this one you abbreviate A for acid, and this one you abbreviate AG for acid and gas production. And of course the tube could be negative, meaning the sugar is not fermented and no gas is produced. Any question about the carbohydrate fermentation tube? All right, the next one we're doing is motility. Some bacteria produce a flagella and they are motile. If they don't lack the flagella, they are non-motile. And if the bacteria is non-motile, the bacteria will only grow in the stab in the motility media. This is a semi-solid media with a consistency of, thin jello, so it's jello and it wobbles. But the bacteria, if they are motile, meaning they have a flagella, they will grow out of the stab, and you see how it's the turbid everywhere in the tube. It's not clear like on the side here, and that's because the bacteria have swam everywhere in the tube, and it has made the tube entirely turbid. The stab was like that, and the bacteria grow everywhere in the tube because they're motile. Any question about the motility test? All right. The next test is the hydrogen sulfide production test, and some bacteria can utilize sulfide ions in the test and react with that to produce, usually with iron, in the tube to make a black precipitate. And so if black happens in the hydrogen sulfide test, then it'll turn black. And if the hydrogen sulfide is not produced in the tube will remain its normal color, in this case yellow, and it will be negative. Now note that hydrogen sulfide only happens in the absence of oxygen or where oxygen is limited. So it doesn't happen at the top of the tube. And so your tube could be half yellow and then half black at the bottom. Okay? Meaning that yellow could be much bigger than this is shown. Any questions about the hydrogen sulfide test? Now I haven't told you how you're testing for it, because there's more than one test to test for the production of hydrogen sulfide. the easiest or simplest to explain is triple sugar iron tubes, and this is a slant where you take a needle, also called the inoculating loop, and you put bacteria on the tip of that loop, and then you stab it into the tube, and you should come at least 2/3 of the way down the tube. You need to go down in the tube. If you only stab the very top, you might not see black, because black doesn't happen at the top of the tube. And then when you're done with that, you re-inoculate the tip of the needle, and then you grow it on the slant. The reason for doing it in both places is here we're growing it anaerobically, and on the slant we're growing it aerobically. We're only looking at in this test for the production of hydrogen sulfide, so you'd look to see if it's black in the bottom of the tube, or negative where it's not in the tube. But this could look at fermentation, and I'm not going to discuss that. And that looks like the only tube we're going to look at, but there are 3 other ways to look at hydrogen sulfide. You could look at it in a Sims tube, which it also turns black, and you can look at it in the HE agar plate, where the colony is black, or it's black right around the colony, if the bacteria produces hydrogen sulfide. I guess I have to talk about triple sugar iron tube, because you're learning about this in this lab, and that is the slant has only 0.1% glucose in it, the slant and the bud, but 1% lactose and 1% sucrose. So there's 10 times more lactose and sucrose than there is glucose. And that's important in interpreting the test, which I'll talk to you about in a minute. It has a pH indicator in it that allows you to determine the pH of the media, as well as peptones for a source of nitrogen, and proteins for carbohydrates, not carbohydrates, for the bacteria to metabolize to get their carbon source. That's what I wanted to say, as well as their nitrogen source. And then glucose, lactose, and sucrose as possible carbon sources, as well as thiosulfate and ferrous sulfate, and that's to detect the hydrogen sulfide production. I mentioned how you inoculate these tubes. You stab it, as well as inoculate the slant. And then you look at the coloration of the growth in the butt and on the slab. So you're looking at the growth where it's anaerobically, and then where it's aerobic. All right, here's some results. This is a negative tube where there's no fermentation aerobically, meaning on the slant, and no fermentation in the butt. Here, this is looking like it is no fermentation on the slant, and then fermentation in the butt. And you can't really see the yellow here, but if it's black, you assume that it was yellow before the black hid the yellow color, meaning you only get hydrogen sulfide production where there is fermentation of the sugar. Because the slant is red, I should probably talk about this one last instead of second, and the butt is fermented, we say that what happened is that initially, if we looked at the slant, it would have been yellow, and the butt actually is yellow. It's just you can't see it because it's black. And so what happened was that the glucose is the only sugar that's being fermented. And once the glucose has been used up, the bacteria use the protein, which makes it basic, and the basic color or the basic ions remove the acid ions, and that's why it's looking pink. So all of the glucose has been used up, but glucose was fermented, and that's why it is red on the top and a yellow on the bottom. The reason why it's yellow on the bottom is because the fermentation happens slower in the absence of oxygen than in the presence of oxygen, and so all of the sugar was used up aerobically but not anaerobically, and I know that's complex, so you can always look at the table for help for identifying this. Any questions about that? All right, so let's move on to the third tube. We note that it is yellow in the butt, but pink on the slant. So once again, only glucose is fermented, and initially if we would have looked at this earlier, this would have been yellow, but when all of the glucose is used up on the slant, the bacteria start digesting protein, which lowers the pH, raises the pH, and then makes it basic here, but there's still some glucose here to be metabolized, and so this remains yellow in color. And the reason is that this bacteria is growing slower because it's anaerobic growth, and this is aerobic growth, and so it's growing faster. And so we say only the sugar glucose is fermented, and the sugar glucose is fermented in both positions, okay? If it's yellow in both the butt and the slant, then we know the sugar glucose is fermented because that's the base sugar, it's always fermented. And then at least one of the other sugars are fermented. It could be lactose, or it could be sucrose, or it could be both of them. Okay, any question about that? And the reason why the slant is yellow here and remains yellow is that sucrose and lactose is present, where do I have that? At 10 times higher the concentration than glucose. And so the sucrose and lactose have not been totally used up by the time limits of this test. You can also look to see if there's gas production, and you can see tube 4 had the production of gas. So you can look at the production in the TSI tubes for hydrogen sulfide. You can look at the utilization of only glucose, and you can look at the fermentation of glucose and at least one other sugar, maybe both other sugars, as well as the production of gas. All right, I'll always provide this table if you need to interpret the TSI tubes. I think that's correct. I'll check on the quiz there, make myself a note. And I provide the table for you for this test because it's so complex. All right, so we mentioned that one way to detect hydrogen sulfide production is with the SIM tube. SIM auger is inoculated with a needle, and you simply stab straight down, going at least 2/3 of the way down the tube. Let me show that. Hope I don't show that. To the bottom of the tube. And if there's black in the tube, you're producing hydrogen sulfide. In the SIM media, you can also look to see if the tube has growth only in the stab, in which case it's not motile, or if it's growing out of the stab, like in this tube here. So we can see that this whole region has growth in it. Let me see if I can go down. Nope. So this whole region has growth in it, and so this is a motile organism. And then in the SEM tube, you add Kovacs reagent, and if it turns pink or reddish, it is positive for indole, and if it's not pink or reddish, then it's negative for indole. So the control is negative, and then tube 5 is negative for indole. This tube is negative for motility. I think that tube is negative for motility. That tube is positive for motility. And this one, it's hard to say, but because the black is everywhere, we say that the bacteria have gone everywhere, and so tube 1 is motile. Make sure I got that. Yeah, motile, indole positive, sulfur positive. This is the only one that's sulfur positive, the only one that's black. All right, any questions about SIM test? For the McConkey's and HE test, it's a selective and differential media. If the bacteria can grow, they're most likely gram-negative. If they don't grow, most likely gram-positive. You do need to check on that, because every once in a while you'll have a gram-negative that won't grow on these media, and you'll have a gram-positive every once in a while that will grow. But over 95% of the bacteria that grow on these plate are gram-negatives. And then you look for their differences in the species, for their lactose metabolism. On McConkey's, if they ferment the sugar lactose, they're pink. If they don't ferment the sugar lactose, NLF means non-lactose fermenter, LF lactose fermenter, they're not pink. This one is off-white or something like that, or white. On the HE plate, the lactose fermenters are pinkish. The non-lactose fermenters are white. If they produce hydrogen sulfide, there will be black colonies on the HE plate. All right, any questions about McConkey's and HE? So you're going to be doing the lab. And you'll be watching video clips on the carbohydrate fermentation test, the SIMS test, the MRVP, and that's really 2 tests, the TSI test, and the McConkey's auger. You'll also need to view some of the website Microbuzz. Click on the differential test link. It's on the left of the screen to expand the menu. You'll only need to click on the test. You'll need to complete the table in step 1 of worksheet to lab 9. Let me see if I've got the worksheet here. I don't think I have it here. Nope. Come on. So you have to answer these questions, what the positive result looks like, what the negative result looks like for the oxidase test, the indole test, and all of these tests over here. You'll need to look at Microbuzz for help on some of these. Some of these, the videos will show you, as well as the lab has discussed them. But where the lab hasn't discussed them, you can look on Microbuzz and then say what the positive result looks like for oxidase. The cells or the spot turns blue. The negative result, I'll give you the answer, it's not blue, okay? And you have to fill in this table for your worksheet. Any question about that? Let's move back. So fill in that table and complete worksheet to Lab 9. You also need to complete the case studies, and I've got the case studies below. Case study 1 shown here, which a person has come in with an infection. They cultured it on blood agar plate, and it looks like McConkey's. And then there's the Gram stain. And the day 2 test results, they did a SIMS test, an MR test, a VP test, the citrate test, and the triple sugar iron test. And then identify the bacteria species which is causing the infection in the patient. And then you just go on for all the other case studies. There are, it looks like 5 case studies. All right, any questions on what you're doing? If not, go ahead and work on the lab. I will be here till 8 o'clock, maybe a little bit later if there are questions, and I will help you with this lab. This lab will take a little bit longer time than other labs because the case studies take a little bit of work. You won't be able to answer this in just a few minutes. It'll take, I don't know, 5, 10, 15 minutes to answer. Okay? All right, get to work. I'll be here for questions. I might grab a bite to eat, but I'll bring it here, so I will be back until at least 8 o'clock. Any questions?
I got a question about work lecture 8, Professor.
So you're talking about the lecture? Let me go there.
Yeah,
I'm getting there, just give me a minute. The rest of you can work on the lab. So this is genetics we're talking about. Actually, I can just go here. Let me go ahead and stop the video. I'll turn back the share in just a minute. So the question was what to do about worksheet lecture 8B or any worksheet, and what happens if all the questions are answered. If all the questions are answered, then just take a question, preferably one that you think somebody might have answered wrong, and answer it. But as long as you answer a question, that's all I'm going to be looking for. Okay? So it's that simple.